Thank you, Dr. Lee, and good to meet you online. It's a great pleasure to make a presentation for AbTis today. We are located in Korea, and the main office is located in Suwon, nearby Seoul City. And we have a Seoul office too. And we were created in 2016. We are focusing on the ADC new therapeutics development. And it was created in about 4.5 years ago. Our core technology is ADC platform technology with stable and cleavable linkers and applicable native to commercial and novel antibodies. So there's no mutation, no engineering necessary to antibodies. And also we have the site-specific conjugation. So it's applicable to therapeutic ADCs and also companion diagnostics and long-acting biopharmaceuticals in the future. The founder and current CEO, Dr. Chung, is also a professor at Sungkyunkwan University in the pharmacy department. And he has that experience at Harvard and Scripps in the background. And also Dr. Choi is a CSO. He is a medical doctor and also PhD. Graduate from Yonsei University Medical School and has experience in Merck, GSK, and LG. So we have a strong management team, as you can see. And also we have strong SAB members, Dr. Joel Snyder, and also famous, well-known scientists and professors and medical doctors. We are founded in 2016 as Seoul BioHub. And we got the funding in 2018, Series A, and Series B actually recently closed early this year. And during that time, we have been completing our ADC linker technology, and we believe it's the best technology, ADC linker technology in the world. We have a strong investor basis, leading venture capitals in Korea, Stonebridge, K2, and also asset management companies like Timefolio. We are striving to achieve our goal through our open innovation policy. We have already have several partners for the co-development of our ADC pipeline together, and also looking for licensing in and licensing out partners throughout the world. Our key technology is called AbClick, and it's the world's best site-specific ADC linker, we believe. And it has two simple, clean steps compared to the other ADC platform technologies. And linker conjugation and drug conjugation in 2 steps, and we have a complete ADC product. I'll explain this technology more in a later part. And compared to the native antibodies, our AbTis ADC has FCR and binding maintained, and biological half-life maintained, and similar circulation and excretion. We have also recently published peer-reviewed paper. Many of you well know about the ADC, but I'll go briefly explain about the ADC. And it has 3 components: antibody, payloads, and linkers. And the market has been really growing fast in the past couple of years. Since 2019, 5 ADC products have been approved by the FDA. So we have a total of 10 products available these days. And as you can see, the market has been growing very fast, especially in the approval of NR2 by Daiichi Sankyo recently is expected to be the blockbuster products in a short time. And also Daiichi Sankyo also had a multi-billion-dollar deals with AstraZeneca, totaling more than $6 billion each. And also global CDMO companies like WuXi and Samsung and Lonza has a strong ADC business also with ADC biotech companies. As you can see, Daiichi Sankyo has the NR2, and they have ADC pipelines, and the stock prices of Daiichi Sankyo is growing quite rapidly in the past couple of years. And ADC has been in a very hot area, especially in the second half of last year, like Gilead acquiring Immunomedics with a 108% premium. It's a total of $21 billion US. And Merck made an equity investment and also has a licensing deal. And also Merck acquired BioCentury. And also Boehringer Ingelheim acquired MBE Therapeutics. So ADC is a very active in investment wise also and capital market wise. So there are big interest in ADC companies. So to explain our technology and our company, we call ourselves as a third generation ADC technology company. And the first generation ADC company is KESALA, which was produced by Genentech, Roche, also AbTis by Seattle Genetics. They are in the market and they are being sold very well actually these days. KESALA and AbTis have problems, have issues because they have a random conjugation. As you can see on the left side of the pictures, you can see the payload and the linkers are conjugated to the antibodies in the random ways. So it is impossible to control the drug antibody ratios too. So it is non-specific conjugation, and so it has problems of inconsistency of a drug antibody ratio too. So it has problem about production wise, obviously because of the random conjugation of that characteristic of first generation ADC. To solve the problems of first generation ADC, second generation ADC companies recently have been developing site-specific conjugation. So you can put the linker and the payloads on the specific side of the antibodies. So the advantage of this technology is that you can site-specifically conjugate your payload and link the linkers on the specific part of the antibodies. So you can control the drug antibody ratio, and you have advantages about that. But the problem with this technology, we believe, is that you have to mutate, you have to engineer your antibodies to achieve that site-specific conjugation. So there is also we believe there's might be some possibility of immunogenicity due to changes on antibodies. As you can see in this picture of rabbit IgG1, there are many lysins and many cysteines are there on these antibodies. So it is very difficult for the site-specific conjugation. Our third generation ADC has advantages over the first and second generation ADCs. We can control drug antibody ratio, new mutation or modification on antibodies are necessary, and site-specific conjugation to lysine in certain part of lysine on antibodies in Fc domain. So it's a Fc binding peptide linker is conjugated, and also it's a traces linker. So our advantages are like drug conjugation to unmodified antibodies. This is applicable to the therapeutics antibodies with no mutation. And this means also very cost-effective ADC production possible. And site-specific conjugation is a covalent binding to Fc domain and high yield of conjugation and simple process of purification. And this means CMC advantages of our ADC development. And the payloads diversity are also available. We can control the DAR, drug antibody ratio. We have 2 and also 4. And more numbers are also possible too. And also, the linker itself is very stable in serum, and it will be cleaved by specific protease within when the ADC gets into the cancer cells. As I mentioned earlier, we have a simple 2 steps for the ADC conjugation and ADC development. Site-specific linker conjugation to antibodies through specific part of lysine, and it's a non-covalent binding interaction by molecular recognition, and then the covalent bonded Fc binding peptide to lysine on the antibody with click functional group. And the second step, the last step, is addition of payloads, drugs to the linkers by toxin conjugation by click reaction. It's a simple 2 steps, and no changes, no mutations on the antibodies are necessary, which is our biggest differentiation, strong points about our ADC platform technology. We have a strong IP strategies. We use US patent farms, and also we use Korean patent farms. This is our 3 major pipelines. We strive to be best in class in the therapeutics development. Our first pipeline is targeting gastric cancer, and we are planning to get into the preclinical earlier this year, and also getting the IND filing later next year. This is our whole pipeline at this time. As I mentioned earlier, we are striving to have open innovations through our partners. As you can see, we have antibody companies like iProgen, SCG Medical, and AbTis. They provide their antibodies, and we provide our ADC linker technology. Together, we can create new ADC pipelines together. And also, we have our own pipelines. The first one is using the Herceptin HER2 targeting antibodies and new payloads. This is the animal study data, and we are trying to prove that our linker technology is very feasible and viable. And it shows that it has the proof that our anti-cancer effects of our ADC using Herceptin DM1 payload with a drug antibody ratio of 4 is working. So in the future, our technology has also applications because of using our platform technology. We are not only developing a new ADC, we also is planning to have an in vivo radioactive imaging platform with on the radioisotope payload, and a long-acting biopharmaceutical platform is also possible using peptides or protein drugs. And lab on a chip is also possible using our technology. We had a meeting with one of the 2 biggest semiconductor companies in Korea, and also they are interested in a lab on a chip using our platform technology. This is our corporate milestone, and we recently closed our Series B with a strong investor support, and we are getting to the preclinical trial this year and IND filing next year, and for the phase 1 clinical trial in 2023. And also we are planning to go for the IPO using the technology exception track in Korea. And the last word is we have a strong ADC platform technology, and then we have a strong management team, and also our pipeline. We have with our partners, so we have a new pipelines also in hand, and thus we have a strong financials with the recent closing of Series D and our strong investors. Thank you very much for listening. Any questions?
Well, thank you for a nice presentation, Justin. Now the audience is now open for question or comment. Maybe you can type your questions on the chatting room or raise your hand. Well, if not, we have a simple question, because you were in the capital market before you joining this company. So you completed the Series B funding very successfully this time. And would you give the market situation currently for KoreaBIO industry?
It is a very easy question, and also at the same time it's a very difficult question. It depends on case by case. I mean, VCs, I mean, I used to be a venture capitalist, so you asked that question, I imagine. And the VCs are looking for the, obviously, the really distinguishable technologies, technologies that is better than compared to the other technologies, have a strong implications in the market and or in the development side of the technology. So our ADC linker technology has that advantages. We believe that venture capitalists believe that, and also strong management is also very important. That the team has been together from the foundation of the company, and also we have Dr. Choi, who is a new CSO. Also, he has been he will play a very important role for our development side too.